Hello everyone. Today we will learn about chromatin, euchromatin and heterochromatin. Since our early days at school, we have learnt that DNA is the hereditary molecule. It lies within the cell nucleus and contains all the information that is passed on from parent to daughter cells. However, this DNA is not loosely arranged in the cell nucleus. In fact, it is bound with nuclear protein called histones and is then organized into a firm compact structure called chromatin. There are specific reasons for packaging DNA into chromatin with the help of histone proteins. The first one is to protect the DNA from damage or enzymatic attack. Second one is to pack the DNA by folding it several times so that the long DNA can be made compact and accommodated into the tiny cell nucleus. Third one is to allow the DNA to be accessed by enzymes for the purpose of replication or duplication of parent DNA for provision to the daughter cells. Fourth one is to allow the DNA to be regulated in its function by enzymes that control transcription of protein synthesis. Thus, chromatin packing regulates how and when a gene should express itself. Fifth one is to enable the DNA to be conveniently separated during mitosis so that each daughter cell gets the correct amount of DNA. Chromatin is therefore a complex macromolecules such as DNA, RNA and proteins which is assembled together. The primary protein component of chromatin are histones that compact the DNA. Chromatin is only found in eukaryotic cells, that is more differentiated cells with well-defined nuclei. Prokaryotic cells have a different organization of their DNA, in which the strand of DNA is localized within the nucleoid region and is termed the prokaryotic chromosome or is called genophore. The primary reason for packing the DNA into a chromatin assembly is to fit the molecule in the much smaller volume of the cell nucleus. To understand this, we must consider that an average human cell contains a very large DNA molecule comprising of about 6.4 billion base pairs of DNA divided among 46 chromosomes. A single continuous DNA molecule is organized into a chromosome. The larger the chromosome, the longer the DNA it contains. We know that each base pair is about 0.34 nanometer in length, hence 6 billion base pairs would constitute a DNA molecule that would be 2 meter long. Now, how can a 2 meter long DNA strand can fit into a cell nucleus only 10 micrometer, that is 1 into 10 raised to minus 5 meter in diameter? The firm packaging and coiling of the DNA reduces its size for this purpose. Secondly, the microenvironment in the cell is replete with nucleases, ions and other small molecules that would unnecessarily bind to the DNA or cleave it. The DNA therefore requires a protective shield, and at the same time this molecule should still be able to carry out its functions. For this purpose, the DNA should be accessible to specific enzymes for replication. It should also be available to small transcription factors and enzymes for transcription. Chromatin is that molecular assembly in which the DNA is accessible to enzymes and regulatory proteins and yet protected from any change in the cell. Moreover, if we just try to imagine an incredibly long string of DNA which is loose within the cell, it would get knotted and entangled within itself as well with other molecules in the cell. However, the organization of the DNA with its nucleoproteins in the form of chromatin prevents any untoward interaction with the DNA and yet enables the molecule to carry out its functions smoothly. The structure of chromatin attains its firm degree of binding from the fact that the nucleoprotein histones are positively charged. Strongly basic proteins, while DNA due to its phosphate groups is more negatively charged. The two molecules therefore bind with electrostatic interaction and other non-covalent forces. The chromatin structure also depends on several factors. The overall assembly depends on the stage of the cell cycle. During interphase, the chromatin is structurally loose to allow access to RNA and DNA polymerases that transcribe and replicate the DNA. When the DNA does not have to replicate or be transcribed, the chromatin is more compact and condensed. This enables formation of chromosomes during phases where the cell has to divide. Hence, in a compact, well-organized structure, the DNA is able to move more systematically to each daughter cell. If the DNA were not compacted and assembled into distinct chromatin-derived chromosomes, can you imagine how long loose strands of DNA could be separated from parent to progeny cells? Much of the DNA would be entangled into a non-functional knotted mass. According to biochemists, the operational definition of chromatin is the DNA, protein, RNA complex extracted from eukaryotic lysed interphase nuclei. Chromatin is the product formed from the combination of DNA and other types of protein. DNA, organized as chromatin, is found two forms along the length of the chromosomes that reflect the level of activity of the cell. First is euchromatin and second one is heterochromatin. These two forms are distinguished in a cytological manner, dealing with how intensely each form is stained. The euchromatin is less intensely stained as compared to heterochromatin. Now we will see euchromatin. When certain dyes like carminic acetic acid, GEMSA or orcine are used to stain certain the chromosomes, certain regions of the chromosomes show up with differences in their staining intensity. The resulting pattern shows alternatively light and darkly stained regions, which are characteristic for the particular chromosome of a species. During interphase, the chromosomes are not distinctly assembled. After mitosis has been completed, most of the chromatin, which is in highly compacted mitotic chromosome form, return to its diffuse interphase condition. Approximately 10% of the chromatin, however, generally remains in a condensed compacted form throughout interphase. This compact, densely stained chromatin is seen at the periphery of the nucleus. Chromatin that remains compacted during interphase is called heterochromatin to distinguish it from euchromatin, which returns to a dispersed state. The intensity of the nuclear staining becomes feebler and less uniform than that of the chromosomes seen at metaphase or anaphase. Heath in 1929 observed the distinct light and dark stained zones, and he was able to distinguish between heterochromatin and euchromatin. All the intensely stained domains were termed heterochromatin, while euchromatin referred to the diffuse or lightly stained regions. Heterochromatin is usually spread over the whole nucleus and has a granular appearance. It is known today that the heterochromatic domains are those where the DNA is tightly packed, that is strongly condensed, which is the reason for their more intense staining. The euchromatic domains are less tightly packed and hence pick up less stain. Most of the genome within the cell nucleus, that is 92% of the human genome, is euchromatic. Euchromatin is a lightly packed form of chromatin, that is DNA, RNA and protein, that is enriched in genes and is often but not continuously under active transcription. Euchromatin is dispersed and not readily stainable. It is prevalent in cells that are active in the transcription of many of their genes, while heterochromatin is most abundant in cells that are genetically less active or not active. Euchromatin comprises the bands which appear as light colored bands when stained with Gemsa stain, that is G banding technique, and observed under an optical microscope. In contrast to these light bands, the heterochromatin stains darkly. Electron microscopic observations have revealed that the basic structure of euchromatin is an elongated, open 10 nanometer microfibril. Though it is lightly packed in the form of DNA, RNA and protein, it is definitely rich in genes and is usually under active transcription. The standard structure of euchromatin is unfolded, elongated, about the size of a 10 nanometer microfibril. This minute chromatin functions in the transcription of DNA to mRNA products. The gene regulatory proteins, including the RNA polymerase complexes, are able to bind with the DNA sequences due to the unfolded, non-compact structure of the euchromatin. When these substances are already bound, the transcription process begins. The activities of the euchromatin aid in cell survival. Now we will learn about heterochromatin. In prokaryotes, euchromatin is the only form of chromatin present. There is no heterochromatin. This suggests that the heterochromatin structure evolved later along with the nucleus, possibly as a mechanism to manage the increasing genome size. Heterochromatin thus appears as a small, darkly staining, irregular particles scattered throughout the nucleus or accumulated adjacent to the nuclear envelope. On the other hand, heterochromatin is a tightly packed form of DNA. It is commonly found on the peripheral areas of the nucleus. According to some studies, there are probably 2 or more states of heterochromatin. Inactive satellite sequences are the main constituents of heterochromatin. Heterochromatin is responsible for gene regulation and protection of chromosomal integrity. These roles are made possible because of the dense DNA packing. When 2 daughter cells are divided from a single parent cell, heterochromatin is usually inherited, which means that the newly cloned heterochromatin contains the same DNA regions, which results in epigenetic inheritance and leads to the development of different levels of gene expression. Heterochromatin is termed as genetically inert, indicating that it has little genetic activity. When a radioactively labeled RNA precursor such as 3H-uridine is given to cells that are then fixed, sectioned and autoradiographed, the clumps of heterochromatin remains largely unlabeled, indicating that they have relatively little transcriptional activity and hence take up or use very little of 3H-uridine. The state of a particular region of the genome, whether it is euchromatic or heterochromatic, is stably inherited from one cell generation to the next. Heterochromatin is said to be late replicating. It begins replication much later than euchromatin in the S phase and continues replicating long after much of the euchromatin has already replicated its sequences. Heterochromatin is divided into 2 classes. First is constitutive heterochromatin and second one is facultative heterochromatin. First we will see constitutive heterochromatin. Constitutive heterochromatin remains in the compacted state in all cells at all the times and thus represent DNA that is permanently silenced. In mammalian cells, the bulk of the constitutive heterochromatin is found in the region that appears near the end of the telomeres and centromeres, that is, flanking sequences of each chromosome, and in a few other sites such as the distal arms of the Y chromosome in male mammals. Hence, it is a major part of the constitution or structure of the chromosomes, and so it is referred to as constitutive heterochromatin. The DNA of constitutive heterochromatin consists primarily of repeated sequences often non-coding sequences. These regions have relatively few genes. In fact, when genes that are normally active move into a position adjacent to heterochromatin, that is, having changed position as the result of transposition or translocation, they tend to become transcriptionally silenced, a phenomena known as position effect variegation. It is thought that heterochromatin contains components which regulate the expression of neighboring genes. The influence of such chromatin can spread outward a certain distance, affecting nearby genes. The spread of heterochromatin along the chromosome is apparently blocked by specialized barrier sequences, that is, boundary elements in the genome. Constitutive heterochromatin remains condensed and does not decondense to go back to the euchromatin state. It also serves to inhibit genetic recombination between homologous repetitive sequences. This type of recombination can lead to DNA duplications and deletions. Now we will see facultative heterochromatin. Unlike the constitutive variety, facultative heterochromatin is chromatin that has been specifically inactivated during certain phases of an organism's life or in certain types of differentiated cells. It thus appears to be functional in nature, perhaps condensed or heterochromatinized for a specific purpose. At given times, therefore, it decondenses and reverses to its euchromatin state. An example of facultative heterochromatin can be seen by comparing cells of a female mammal to those of a male. The cells of males have a Y chromosome and a much larger X chromosome. Because the X and Y chromosomes have only a few genes in common, males have a single copy of most genes that are carried on the sex chromosome. Although cells of females contain two X chromosomes, only one of them is transcriptionally active. The other X chromosome remains condensed as a heterochromatic clump called a Barr body after the researcher M.L. Barr, who discovered it in cells of the female feline species in 1949. Formation of a Barr body ensures that the cells of both males and females have the same number of active X chromosomes and thus synthesize equivalent amounts of the products encoded by X-linked genes. This phenomena was called dosage compensation. The British geneticist Mary Lyon studied X chromosome inactivation and based on her findings of the inheritance of coat color in mice proposed the following theory in 1969, which came to be known as the Lyon Hypothesis. First is heterochromatinization of the X chromosome in the female mammals occurs during early embryonic development and leads to the inactivation of the genes on that chromosome. Second one states that heterochromatinization in the embryo is a random process in the sense that the paternally derived X chromosome and maternally derived X chromosome stand as equal chance of becoming inactivated in any given cell. Consequently, at the time of inactivation, the paternal X can be inactivated in one cell of the embryo and maternal X can be inactivated in a neighboring cell. Once an X chromosome has been inactivated, its heterochromatic state is transmitted through many cell divisions so that the same X chromosome is inactive in all the descendants of that particular cell. Third one is reactivation of the heterochromatinized X chromosome occurs in germ cells prior to the onset of meiosis. Consequently, both X chromosomes are active during oogenesis and all of the gametes receive a euchromatic X chromosome. The Lyon hypothesis was soon confirmed because although maternally and paternally derived X chromosomes may contain different alleles for the same trait, adult females are in a sense genetic mosaics where different alleles function in different cells. X chromosome mosaicism is reflected in the patchwork coloration of the fur of some mammals. The random inactivation of X chromosomes discussed here, which occurs after the embryo implants in the uterus, is actually the second wave of X chromosome inactivation to occur in the embryo. The first wave, which occurs very early in the development, is not random but rather leads only to the inactivation of X chromosomes that had been donated by the father. This early inactivation of paternal X chromosomes is maintained in the cell that give rise to extra embryonic tissue, example the placenta, and is not discussed in the text. Early paternal X inactivation is erased in cells that give rise to embryonic tissue, and random X inactivation subsequently occurs. Euchromatin and heterochromatin have distinctly different properties. A comparison of these two forms of chromatin helps to understand the differences in these two forms of chromatin with respect to their function. First one is, chromatin makes up the nucleus. It is made up of DNA and protein and has two forms, euchromatin and heterochromatin. Second one is, when stained and observed under optical microscope, euchromatin is the light colored band while the dark colored band is heterochromatin. Third one is, darker staining indicates tighter DNA packing. Heterochromatin thus has a more compact DNA packaging than euchromatin. Fourth one is, heterochromatin is termed as genetically inert and has no genes encoding for proteins, while euchromatin is characterized by coding genes. Fifth one is, although termed inert, heterochromatin plays a role in the regulation of gene expression. Sixth one is, heterochromatin is a compactly coiled region while euchromatin is loosely coiled. Seventh one is, euchromatin contains less DNA while heterochromatin contains more DNA. Eighth one is, euchromatin is early replicative while heterochromatin is late replicative. Ninth one is, euchromatin is found in eukaryotes, cells with nuclei, and prokaryotes, cells without nuclei. 10th one is, heterochromatin is only found in eukaryotes. The functions of euchromatin and heterochromatin are gene expression, gene repression, and DNA transcription. Let us summarize the topic chromatin. Chromatin is a complex of DNA and proteins that forms chromosomes within the nucleus of eukaryotic cells. Chromatin in the nucleus exists as euchromatin or heterochromatin. Euchromatin comprises the most active portion of the genome within the cell nucleus. Major portion, that is 92% of the human genome, is euchromatin. The remainder is called heterochromatin. Euchromatin is a lightly packed form of chromatin, that is DNA, RNA and protein, that is enriched in genes and is often but not always under active transcription. Euchromatin participates in the active transcription of DNA to mRNA products. Heterochromatin is a tightly packed form of DNA which comes in multiple varieties. These varieties lie on a continuum between the two extremes of constitutive and facultative heterochromatin. Heterochromatin has been associated with several functions, from gene regulation to the protection of chromosome integrity. Some of these roles can be attributed to the dense packing of DNA, which makes it less accessible to protein factors that usually bind DNA or its associated factors. Thank you.